Longest common subsequence. The longest common subsequence, LCS, problem amounts to find the longest string that is a subsequence of every string in a given set of strings. The subsequence here is not composed of consecutive letters in the string, but it can be achieved by deleting some of the characters from the string. The problem can be solved polynomially in the case of two strings, but in general, the problem is NP-hard. The example of what the LCS problem is explained in Figure 3. The example of the LCS problem. For a given set of strings equals, katigak, atiekgatak, gategak, agicht, the longest common subsequence of each string is atgak. The LCS is underlined in every string. The LCS problem is used in bioinformatics to compare sequences of molecules, DNA, RNA or proteins, in order to find homologies between sequences of organisms of different species. The homology helps to predict the function of unknown genes. If their sequences are similar to those of known genes, one may expect that the function is similar. The other applications of LCS can be found in text editing or data compression, among others. Tabatabai and Musavi proposed a hyper-heuristic for the LCS problem. A beam search algorithm in its standard form is a tree-based procedure. It starts from the initially empty solution and extends it by one letter in every iteration. All possible characters sigma are evaluated by a function f. As possible extensions, but only beta best ones are further explored in the next iteration, beta being the size of the beam. A hyper-heuristic approach is introduced here to choose the best function f. Among the two available, Power-on-Prob. Power takes into account the length of possible suffixes, with the high impact of the minimum suffix, after deciding on the possible extension character. Prob, on the other hand, calculates the probability of a random string being a subsequence of the suffix. The hyper-heuristic runs the beam search algorithm twice in every iteration with Power-on-Prob as f. Function and chooses the one which gives the possibility of a longer subsequence extension. The method was tested on random biologically inspired and real sequences of DNA and proteins of rats and viruses, sigma equal to 4 in the case of DNA sequences and 20 in the case of proteins. Hyper-heuristic appeared to superior to the beam search method used with just one heuristic, either power or prob. The proposed hyper-heuristic in comparison with the state-of-art algorithms MLCS app and DEA, depending on the tested dataset, provides 1 to 2% and 19 to 25% improvements in the solution quality, respectively.